So let's go ahead and get started. Let's get started. Welcome. Nicola has been here since Monday and have had meetings after tagging it on the IGSS. So we're going to get like the deep dive into this excellent paper. Yes, thanks very much.
Thank you. Yes, thank you very much. Thanks, Jason, for the invitation. It's been a very nice visit. I had the pleasure to meet with some of you already and discuss a bit research ideas and so on. So it's a very nice visit. So what I'm going to present is it's a paper that we have been working on this paper for a while now. It's a pre-pandemic paper that's stayed a bit. So we're trying to now to find a way to finish it, and I'm very eager to know about your if you have subjective opinions to discuss with you. And also because I know that it's a group that it's familiar with all genetic terms. I'm not going through what is PGS, Polygenic score, PGI, and so on. So the outline for this talk is this. So I'm going to start talking about fertility behavior in the UK, in particular in the cohorts in the of 1940s, 50s, and 60s, because our data are here for these people, but also because we think that there's an interesting demographic dynamics related to fertility that are around this cohort. Then I will talk a bit about our measurements of genetics and environment. This is a paper in which we try to look and understand a bit of the gene-environment interactions, and also talking about a few problems we came across with this measurement and so on. And then we're going to switch to an analysis by cohorts, looking at genetics and different influences across different cohorts. Cohorts that, as we've seen a minute, experience dramatic change in fertility behavior. And then we're going to switch to a particular change. We think that it's very relevant. It's the role of oral contraception, the pill, contraceptive pill. And then we try to play a bit to understand what this G by E interaction means. Okay, so the UK Biobank, that's the data that you probably, many of you are already familiar with. It's covers individuals that are born between 1938 and about 1968. And this is a period in the UK of dramatic change in fertility. So the number of children went dramatically down, as in many other Western countries. And then the increase of women who remain childless has increased a lot. These are women that are older than 45 years old, so that we can presume that they will not have children. And so we pass from about just slightly more than 10% for the cohort born in early 1940 to more than a quarter, 25%. So more than a quarter of women. Yes.
So how is this childlessness defined? It's like women get married but have no children, or they just didn't get married?
Yeah, this is a simple indicator of 1 if you ever had a child, and 0 if not. So it doesn't say if these are voluntary childlessness or involuntary. It doesn't say if it's infertility and so on. This is a trend in many European countries we observe. Yes.
Does this reflect the women of all ages, or it's just over 45?
Older than 45. So it's a standard way to look at, if you want to look at cohort fertility, and also the number of children, by the way, we restrict, and that's why we don't you see the line doesn't go up to 65, so these are women older than 45. And as a convention, 45 is the age limit for which you can look at cohort fertility.
It is surprising that there wasn't an increase after the war. Previous slide, I guess.
Were born in this birth date. So yeah, and it's so people up to here experience the
Oh,
war.
I see. So this is number of children of people who were born in that.
Yes.
Okay,
yes.
I see.
Yeah. So in demography, it's standard to have measurements that are measured in a particular calendar year. So, and the measurements that are referred to the cohort. So these are cohort specific data. So yes, more than a quarter of women born in 1960s didn't have children. We don't know if by choice or for other reasons. Obviously, this also reflects increase in women labor force participation and change in gender attitudes. I mean, there's a huge cultural revolution, if you want, in the 60s, from the 50s and the 60s in the UK. So there's also an increase, almost steady increase of age of first birth that went from an average of 24 and a half years old to 28, and it's increasing. And what is actually very interesting to me is that the age of first sex has declined conversely. So if you think for people born in the 1940, the average age of first sex was 21, and 24 was the average age of first birth. So the interval between being sexually active and having a child and probably marrying, we don't have a marriage information. This data was 3 years. So for the later cohort, we go from 18 to 28. So for the age gap passed from 3 years to almost 10. So huge evolution. This is also referring popular culture as a sex revolution in the UK. You can imagine the Spring in London and the Austin Power moving. So all the stuff. Maybe some of it too young. Okay. What's also interesting is that those were averages, so the average age of first birth, the number of children, and so on. But we can also look at the variation. So here we take the coefficient of variation, that's the standard deviation divided by the mean. And we see that there's an increased variability in how many children people have. There's also an increased variability on the age of first birth. So if this means that back in the 40s, people born in the 40s, they had lower age of first birth, higher number of children, but it was more concentrated on around the mean. And there's more dispersion among the later cohorts. And this is for age of first sex. It's numbers and the scale is a bit different here, so a bit unstable and some sort of increase at the end. So we start from this descriptive analysis, and we try to understand whether genetic endowment affect this heterogeneity in women fertility behavior. We start, our starting point are some work I was involved with is on GWAS of fertility behavior, in which we did GWAS on number of children and age of first birth and also age of first sex. And we did some preliminary analysis on heritability by cohort, and we saw some interesting aspects. We try to model this. Try to understand what's the role of gene-environment interactions, and in particular focus on one big change that we think it's very relevant for women born in these years. That's the diffusion of oral contraception. So as I said, I use by UK Biobank. It has genetic information of 500,000 individuals. About half of them are women. And we focus on age of first sex, age of first birth, complete infertility, and childlessness. About the measurement, yes. So we focus on the cohort born 38 to 69. We exclude from the analysis relatives that we identify by looking at genetics. For this paper, we focus only on women. Also because age of first birth is recorded only for women, but number of children, age of first sex is also for men. And for complete infertility and childlessness, we exclude women that at the time of the interview were younger than age 45. The sample size, you will see later in the regression, depends on the outcome. So for age of first birth, you need to have had the child, obviously. So it's slightly and later. So yes, UK Biobank is an amazing data resource for biomedical information. Has many medical tests. He has linked with the HES, that's the health electronic records. It has imaging, brain imaging, and all sorts of stuff. But he has very poor, in my opinion, measurement of the social environment. And so this should spark some sort of reflection that it's not only important to collect as many data as possible or some many medical information, but sometimes some good measurement of the social environment could be useful, and that it's important to work in the discipline. What is interesting and not many data have, though, is some good geolocation. So these are the plotted location of birth of individuals. This reflects a bit of the obviously the density of the population in the UK. That's far less people living in Scotland than in England, and so on. And probably some error of people. Now, this is actually the zero coordinate. Some people that are recorded here, but they're not living. See. It's a 1 kilometer grid. Precision. If you need a special access, you can ask UK Biobank to have more precise, I think 100 meters grid. But you also reflect the fact that data were collected in, if I remember correctly, 12 different locations in which, because it requires extended medical examinations, that people were going to the location. So it's not necessarily representative of the entire population. Moreover, the UK Biobank has quite big problems in terms of representativity, since only 5% of respondents constitute only 5% of the people that were asked to participate. So that's some sort of selection. So also the graph on fertility, you see there's slightly off with respect to the national the statistic of the Office for National Statistics. So there's a bit more of childlessness. There's a bit higher age of first birth. So it reflects. We can probably think that UK Biobank is selected for socioeconomic status and so on. Nevertheless, I think these are important information because we can plot each individual to a specific region. These are the local authority district. The geography of the UK is quite complicated. It's that's obviously England, Wales, Scotland, Northern Ireland. Basically, almost no participants from Northern Ireland. Yes, different regional level. The smallest available, basically, it's this thing that is called local authority district. So we can plot each individual in a local authority district, and we can derive information at the contextual level. So our measure of G, in this latest version of the paper, we decided to use the polygenic index repository. This is an initiative from the Social Science Genetic Association, and it is brilliant because basically they computed polygenic scores for different cohorts based on summary statistics. They had excluded doing meta-analysis, excluding the cohorts and so on. For the UK Biobank, they do MTAG, so they put together different traits, they leverage the ID of different traits, the genetic correlation and the ID structure, and they have quite predictive scores. For the UK Biobank, well, because UK Biobank is essentially the biggest cohort in every GWAS, probably excluding maybe the educational attainment in which they have the 3andMe data and so on. What they did to not exclude the UK Biobank completely, they split the sample in thirds, and they on random third they calculated that they used 2 thirds of the UK Biobank to calculate GWAS and weights, and they use this to calculate scores. The resulting thing is that we have a score for different subsamples, but we can only use 1/3 at a time. So we use the scores for educational attainment, age of first birth, and number of children. And we didn't use for age of first sex because it's not in the PGR. We should do it ourselves with the same methodology. I know that I shouldn't have said this anymore after the IGSS, but for working on a homogeneous sample, we restrict to individuals to European ancestry and exclude relatives. So this is our actual measure of G, but in the working paper that is out, we use another strategy. In which we I choose to show this slide because I know some of you are also using split sample strategy. So here we did basically the same thing with the 10 different samples, but we put together the results later to have a score for everyone. And it turns out that if you, I don't know how many of you are familiar with this paper of Becker et al. The researchers, so they write in the paper, researchers may decide to conduct analysis that simultaneously include individuals from different partitions of the data, or to meta-analyze the results across different partitions. What the authors show is that such analysis will produce estimates that are unbiased, but the standard error will be incorrectly calibrated. So they recommend that the researcher only do analysis on certain individuals within a partition, or they should include the caveat that the standard error may be calibrated. But so for this reason, we talk a lot that our results, the one that are in the working paper, it's the scores are more predictive. They are based on, we don't know exactly because we have a larger sample size, and also the GWAS, the resulting GWAS was based on a larger sample size. That's the reason why actually that we stopped a bit with the paper, because when we saw that this paper came out, and made us think about was the best strategy. We thought also alternative strategies because of reducing the working sample to 1/3 of the entire UK biobank as all these drawbacks. So we thought one way could be to work with summary statistics without the UK biobank. We have it for the fertility phenotypes. We have it, but often the UK biobank is the largest sample of the GWAS, in particular for things like age of sex. Basically there are not many other samples that have information on age of sex that can be used as a training set for GWAS. We are also thinking, we asked 23andMe to have a summary statistics on 23andMe, so that they should be large enough to Another idea is that to use a summary statistics for gender-specific GWAS. So, for example, using the men GWAS on fertility as so run a GWAS, well, already done on GWAS on age of first birth, number of children, age of first birth on men, and use this as weights for women. So we can use per sample. You might think, okay, but the genetics or male genetics for fertility may be different from women. Yes, we know that there are gender-specific loci, but we also know that the genetic correlation between sexes is almost one, but no different. So these are some ideas on how to overcome the shrinkage of the sample to 1/3.
So that statement on your previous slide, was that just a statement in the Becker paper, or
Yes.
did they provide evidence?
They provide evidence, yes. It's in the methods section of the paper, and it's well, the idea behind that is that since we are using the other sample of the other subsample as weights, there's a correlation structure that we ignored that could affect our estimates in regression framework.
But they did some actual empirical analysis. So that,
they did some. No, they only provide the formula of where the correlation comes from. They do some. I had an exchange with Patrick Turley, and he did some simulation, and he convinced us that there was some. Okay, but now we try to look at. We go back to our descriptive statistics, and we try to see what is the behavior of group or different genetic group. So here we take the top, the individual with a PGI, two standard deviations. The top 5% PGI and the bottom 5% PGI. So we look at the extreme essentially, and we look at what's the behavior across birth, the year of birth. So we also show in the paper that the genetic there's not much difference in the genetic across cohorts. So the PGI are almost stable across cohorts. So this is something due to other factors, and we see that there's a general increase of age of first birth, as we saw on the previous graph, but we see that there's also the divergence between the two groups. So if this distance was about 4 years for people born late 30s, we see that the distance now is almost 6-8 years. So it has increased, and there's a divergences. This we think that suggests some gene-environment interaction, where the environment here is the cohort, the year of birth. We see something similar on a number of children. We see that for the earlier cohort, the distinction is very low and then increases over time. For the age of first, yeah, I don't know, for age of first sex. So we estimated a model in which we have a G by E model in which the E is 5 years birth cohort. And this is the average marginal effect of the PGI by year of birth. So if there was no gene-environment interaction from or gene cohort interaction, we will see a straight line. So the marginal effect of the PGI is the same across all cohorts. What we see, though, is that the PGI seems much more relevant among the recent cohorts for age of first birth. And similar for number of children, we see that the effect is increasing over time. And this is similar to when people do heritability by cohort, seeing that how much genetics explain if there is a differential explanation by cohort. For age of first sex, sorry, the chart, we see that there's if any, there is a bit of a declining effect of genetics over cohort. So this was more important among this cohort. It became less important starting from here. Okay, so now we want to think, yes.
I wonder if, have you looked at some of those trends in ELSA?
Mm-hmm.
Just because ELSA is population representative, like
Yes.
HRS, and they
We should look into that.
you know, because it might be just as a comparison to UKB because of like mortality selection and all these other issues. And all the non-random.
I mean, mortality selection, you have a nice tool.
True, yeah. I guess it's more non-random participation bias, yeah.
I mean, we are confident that this is because so in the GWAS paper we did some heritability by cohort for age of sex and other children. We see that this suggestion of increased heritability by cohort. So this would mean that if you have a good measurement of G, the effect would increase over time. So and the GWAS is not only on the UK, but it's a different cohort. But
And also because I don't know if ELSA was in the original GWAS, but if they weren't, then that was that issue too. Or at least I know they have.
Yeah, I have to figure out how so
I think they have, like the HRS, they have like publicly available PGIs that don't include, if they were in that analysis, they don't include. So you could just look at them. See. I don't know how they were calculated, but
I think the challenge on ELSA will be that your biobank data are like 15 times larger than ELSA, probably by cohort.
yeah.
That you're wanting to.
here you can look almost here cohort. So now we stick to 5 years because we have the 1/3 sample. Yeah. Okay, one thing that we think is relevant for fertility is contraception. I mean, there's huge literature on the role of contraception in that's effect not only on fertility behavior, but also on female labor participation, education, on gender roles, so on. So what about the pill in the UK? So in 1961, oral contraception was introduced in England and Wales, but it was a huge public debate, and it was restricted only to married women. So there were if you read a bit on the history on the contraception in the UK, you have in the 60s that women that go to the doctor pretending to be married or try to get access to the pill from friends that were married and so on. From 1967, the pill is extended to everyone, and it's available through the National Health System or the FPA clinic. So the FPA is a Family Planning Association clinic. It's a association that has been created in the early, I think in the 1920s to promote awareness about reduction, and to reduce fertility in a conscious way. It's a very fascinating story also. And then it was introduced in Scotland and in Northern Ireland. But meaning that from the 67, you could go to these clinics, have some consultation, and they could prescribe you contraception. There are no regional differences. I know in the US, they leverage, some papers leverage the state differences to pill access. It's not possible in the UK. There are two questions, though, in the UK Biobank. The first is, how old were you when you first went on the pill? And the second, have you ever taken a contraceptive pill? So if you plot this age when started using a pill, you see that for the earliest cohort, the pill wasn't available here. So the age drops, and it goes from this is the first cohort, and 49 is the first cohort that has access also for married women cohort. So you see that there is some sort of reduction, then stabilizes from the mid-50s to about the age between 18 and 20. So what we did, we took the geographical information. Yes.
Sorry, yeah. I would like to ask, are there other contraceptive services they can use before the pills are available, like condom or any services?
Yeah, I mean, yes, obviously. So the condom was available and used, but I mean, for contraception has the great advantage that put the woman in charge on deciding when. And so they taking the pill, you can engage in sexual activity without reducing dramatically the risk of child. So I think it reflects also on the age for sex. Yes, people could use condom, mostly condom. I think mostly withdraw, and I don't think the other modern contraception were available at the time, but I can look at it. So we calculate by local authority district the proportion of women who use the pill for the first time and do not have children. We want to have as a reference the women that have not children, and we calculate it by year and district. And we link this variable to respondents where they were 18 years old. So this is a contextual variable. Everyone in the same area will have the same value. And we think it measures some sort of a social norm and social influence. So when you're 18, you have around you women. It measures somehow how available and diffuse is the pill. Obviously, it would be nice to have information on how many pills were sold in that area and some sort of supply measurement. We are kind of working on that, but I think it's a measure that nevertheless it shows some sort of social norm around the pill in that area. So this, there are some geographical variations. So this is by year measurement, not by cohort, but by year. So we reverse it. Or we can also plot in a map. So this is born in 51, then 56, 61, and 67. So you see there is some geographical variation. But most of the variation, and this is an important issue is that most of the variation is temporal variation. Most of the variation in the pill is by, it's on time. Yes.
So you calculate this measure in UK.
Yes.
And is it UKB geographically representative? Like how?
Not quite, because it has some sort of there's a sort of over-representation of people around this district. There is some. We should apply the. There is a paper which calculates weights to rebalance the geographical differences. So basically, he plots some radius around the examination centers and use this information to reweight.
Yeah, because I'm just wondering what.
But there were many centers in many. There were centers around the major cities. So. Yeah.
One question about your norms measure. I'm trying to sort out. So you said it was year by district as the measure, but it's not by the age of the women themselves. It's just like take a year and take a district and you ask what proportion of UK respondents were on birth control.
We did two measures. So one is all women not without children older than 18.
In that year, you mean?
So basically, yes. So the thing is that everyone is asked if they ever used the pill and when they started using the pill. Then we don't know for how long they use it or not. So we have a measure for each calendar year. We have for each person a measure that if has ever used the pill or not. So for each year, we look at people that are older than 18 and ever used the pill in that district. So we have we link this year to we have an information for each. So we have information for each district that is time variant.
That's what I'm wondering is how it is. So take 1956 or something. Anyone who is under 18 in that year, you're not looking at.
Yeah.
Anyone who's older, you are.
Yes.
And you're creating a measure of the proportion of those women
In that year.
Then you take the next year. What are the changes that happen? So you get one more year of people who are now 18.
Well, some people maybe they didn't start using it 18, but they started using it 19.
But
the people that weren't 18 and the previous year that are now.
But is it true that like the biggest part are just people who last year met your criteria and now this year also?
Yeah, okay. So we did another measure in which we restricted to age 18 to 30. We look only at this window of women that were in age between 18 and 30, because here obviously we're capturing also older women, but not many because the cohort, there are not so many in this cohort. So, but we are capturing women that started using very late. No, that they haven't started. So it's moving with the cohort. So we restricted to 18 to 30. So the idea is that, okay, I'm 18, how many women around me that have slightly older than me have ever used the pill?
Is that how you're attaching it to the egos? It's like the year for me is when I'm 18.
Yes.
And it's based on my place of birth and my 18.
Yes.
And then I look at people who are 18 to 30 in that year. But again, if for the people born 1 year later, it's like mostly the same people that you're calculating, right? Except for these people who just turned or are initiators of birth control during that age, right?
I see what you, but it's also we don't have a time variant measurement of the pill.
I'm wondering,
So we know, okay, have you ever used it? So if they ever used the last at age 18, yes, they ever use it also.
So maybe I'll ask, I don't remember what kind of time fixed effects and stuff you're going to engage in, because it seems like a lot of your variation, as you just showed in this next graph is temporal.
Yes.
So like what you're able to control for seems.
Yeah, so no, we okay.
Before we move on, just are you assuming that people still lived at the place of birth?
Okay, that's another issue. So we did some robustness analysis in which so we have the area of birth and also the area of interview. So we calculated people that stay in the same region. So the stayers, we run the regression on the stayers and we have consistent results.
Okay.
But yes, also because
some people would argue this is again biased sample because they are at the same place.
Absolutely, but we don't have the place where they were at age, let's say 18. It's mostly relevant for fertility. So if we take only, and we are a bit hesitant to say, okay, we take only people that were born and lived, and they're measured at age 50 in the same area, because even if they move, if we see that they move to another district, we don't know when they move. So if we think that most of the socialization and decision related to fertility happen when they are in the kid or adolescent, then restricted to only maybe the place of birth is enough. So here, what we estimate, we have an effect for G, where you have a specific polygenic index for the individual I on area A. We have the measurement, the field measurement, the environmental measurement. Then we have an interaction, and then on the fixed effect, we have area fixed effect, localatory fixed effect. Then we include the 5 years for the fixed effect. Then first 20 principal components, the interaction between the principal component and the environment. We class the standard error at the area level because of the environment is measured at the area level. We have also other specification with the year fixed effect and of 5 years fixed effect, because that's very similar. And these are the the results. So yes, we see a consistent effect of genetics. So one standard deviation of genetics is associated with the alpha year of age of first sex, 1 year age of first birth, 9.09 children, and 2% of childlessness. It goes in the direction that we predict. Here for the PGS, we use PGS of PGI on education. What about the environment? We see an effect, the strongest effect we see on age of first birth and number of children. And this means that 1% in our pill availability is associated with 0.03, so it means that the change in 10% is associated with like a third of in age of first birth. We see much less effect on age of first sex. We don't see on childlessness, but childlessness is very similar to the number of children, just that measuring binary outcome. But we see evidence for all 4 traits. So this is 5% star, but the biggest influence is on age of first birth and ability, with much less influence G by E effect on age of first sex. These are the sample size. We estimate the incremental R square, and it's about 2% incremental R squared by adding genes to the model. So we have a model without and with model, and with genes we have about 5% for age of first birth. Also in the GWAS, so that's a trait we can capture genetics. Okay, another way to look at these results, and also to look at that, I think I can help to describe it, is studying the marginal effect. So this is the genetic distribution. What's the effect of the pill, 1% increase in pill across the genetic distribution? For age of first birth, it seems that there's almost no effect on the low genetic propensity, so the people that would have a propensity to have children early in life. This is quite interesting, much higher effect of the pill on people on this side of the distribution. For those who were predisposed, have preferences, attitude on higher fertility. Similar results on number of children. Almost no effect, well, very low effect in absolute value on people that were predisposed to have a higher number of children, much higher on the other spectrum. And this is okay, childlessness, that's similar to fertility, really. And age for sex, we see that especially when we control for cohort effects, effect, we have much lower gene environment interaction effect. The slope is not very steep. If any, however, it seems that the genetic effect is bigger in its absolute value among people who would who would have sex later in life by the genetic disposition. Okay, yeah, we did another. Now we're trying to understand what to make of these results and how to put together the cohort analysis with the regression analysis. And so what we did here was these are the coefficients, the blue one, of a regression model in which we have a cohort interacted by genes, cohort genes and cohort by. And we see the effect, the cohort effect over time. So we see that, yes, there's an increase on age of first birth among later. We do the same model, in which we plug in also the genes, the pill effect, and the genes interacted with pill. So we want to compare the results. And this is, we think it's a kind of naive analysis or moderation analysis in which we see how much the effect, the cohort effect, is moderated by the inclusion of the pill. What is striking and very interesting for us is that when we include the pill in the model, we basically explain all the variation in cohort variation for age of first birth. We also look at the interaction effects, that this is an interaction between cohort and G, and we see that most of the interaction go away when we include the interaction with the pill. So here it's a very naive, we are not estimating how much is moderated, and I know that there are quite some techniques on how to study this. But this is instead the number of children. We see that decreased number of children over time. What happens if we include the pill and interaction, pill interactive with genetics, so that a big part of this cohort effect goes away, but still there's something not explained. That it doesn't go to zero. It means that there is still some cohort variation that is not fully covered by our measure of pill.
But on that one, it looks like that, isn't everything being compared as like 35 to 39 or something?
Yes.
So in some sense, you need to draw a line through all your yellow bars. So you're not able to explain that constant against the one cohort. Is that misinterpreting what you're showing here? So I think you're describing that these yellow bars aren't zero, right? But they're all like the same against the omitted group, which is the earliest cohort.
Yeah.
Right. So in some sense, you can't explain the jump between the first cohort and all the rest, but within all the rest, you can explain it. Is that a correct
I see.
interpretation?
I said because they are flat. Right?
Yeah. And
It
it
seems that there's the first jump we can't explain, but the rest is
Right.
actually.
so it makes me wonder about the first being the weird one rather than the rest.
Okay.
Which is
But
the
it's interesting because of the first cohort that didn't have the pill.
right. But if you switch to your omitted group to be the second cohort, right?
Yeah,
you'd
no,
have,
that's
you'd have a
that's
strangeness in the first cohort, but all the rest of it would be explained essentially.
So maybe it's just the those were the children of the world.
Well, you wonder about if there's then weirdness in the first cohort, that's the explanation rather than the failure that you're thinking this is a failure to explain it.
Yeah, on that great point. Similarly, we look at age of sex, and for age of sex, what is interesting is that we explain very little. The pill doesn't seem to, at least our measurement of the pill, doesn't seem to explain the cohort changes in age of sex.
That's right. With a lot of with your environmental measure and the genetic measures, there was just so much going on for women at this time too, with like labor market, education. And on the environmental side, it seems like you might want to regress out years of education or something when you're constructing that measure.
Yeah.
But then also on the G side, when you want to control for like PA, PGS and these other things, because those are all correlated with these genetic measures of for spurt. Right.
Yeah, so we did a bit of robustness in either side. With the PGI, we included PGI for education, and well, this is actually education, but in the other models, we included PGI for education, for the fertility PGI, and PGI for risk. And as you said, there's a huge genetic correlation between these traits. So, wanted to.
Those are also
Now, way to keep it simple. On the environment, we would like to have an environment that are not individual specific. So, putting an education would, I mean, the choice of how long staying in school is obviously connected with all the fertility choices. So, we actually in a minute we have a contextual measurement of the education. So, yes, I mean, and this is, this is,
yeah.
So we are trying to do with three other different variables. So we estimated some sort of average educational level by cohort and district in the same way we did with PIL. We also got some regional estimate of GDP. These are regional on the county level, district level. We also estimated from the labor force survey returns to the sufficiency of return to education, and trying to see if this is linked with the fertility. And so this is, for example, with the average education, and we doesn't seem to explain as much as we did with the PIL. These are really recent analysis. Actually, it was inspired by the presentation at IGSS on David on sortative mating, which he did a full mediation analysis, and I wanted to see if that could be something useful for our paper. And so we don't see much on our education.
But the measure here is it place of birth based and cohort based?
Yes.
So it's within EAB, you're constructing average of my peers.
Yeah.
And do you always exclude the person's own?
Not in this. But the samples, I mean, they are quite a good sample size for it.
Yeah. You have interesting.
We did also a few simulations to understand a bit what's going on. So let me explain this. So this is the light dotted line. These are the observed values plotted against our baseline model. And since beta is doing good job in predicting. Then we said, okay, if we take this model, what happens if we give to everyone the maximum value of G? So assuming that there's no genetic difference, so they all have the maximum value. We see that the age of first will jump and be almost parallel with the previous line. On the other side, we look at what happens if we give it to everyone the maximum level of the pill. So if pill was fully available for everyone starting from the 40, we see that there would be basically no variation in age of first birth. We did another thing to understand where are these gene environment interaction effects. So we estimate these two large lines are the model with the baseline model. And then we estimate, we predict a value on the same model where we set the gene environment interaction to 0. And we see that basically the big part of the gene environment interaction effect come on the later cohort. So the gene environment interaction effect make because it's all most of the variation is temporal. We see that it's more relevant. The G by E effect are more relevant among the later cohorts. And what are the limitations of this paper and why it took different direction? We don't know exactly where to go. It's well, the pill is measured at the contextual level. This is a limitation, but also a strength in a way because it's not choice of the individual. But nevertheless, even if it's a contextual, it's not necessarily correctly identified because it might be possible omitted variables, so we don't know. Obviously, this is we interpret as a measure of social norms because it has a lot to do with the pill. It's not only taking the pill, but it's the role of premarital sex and society, the gender roles, and there's so many things going on. Obviously, it would be nice to have some sort of exogenous shock, an ID for the pill. We are trying to, we have an array that is the National Library trying to get all the documents of this fertility plan, FPA clinics to see where they were located. And, but they were also at a certain point from 1967, they were incorporated on the NHS, so it's easy to find information, specific information. Other problem, G is also measured with error, so G is it's a proxy of the genetic effect. There might be other effects, so we can include other PGI, or we can have some sort of correction measurement error, exactly how, but and also G is not randomly distributed, so some work in the UK show that if you plot the PGIs on a map, you see that it's higher in the London area, it's lower in the coal mining district, and this reflects not that reflects essentially patterns of sort of mating, migration, and so this could be a problem. Eventually, one preferred doing signal analysis. We did in the previous version of the paper, but now we have some problem in statistical power, especially because when you do signal analysis, the G effect is reduced because further, but also the E, because they are a few years apart. And also it's a result of the G by E effect are very small compared to E. So it's about 1/10 of the effect of the E. So does it make sense to study G by effect if they're so small? I do think so, because especially in the latest cohort for fertility, we see that there is part of this variability in age of first birth in particular, but also number of children could be caused by the genetic diversity. And so to conclude a bit, the genetic differences result in fertility differences, especially in later cohorts. We see the greatest genetic heterogeneity we see for fertility postponement. This seems less relevant in the decrease of for age per sex, so probably something else. For all traits we show G by interaction. If we take for the birth cohort as an environmental measure, we think that introduction of contraception capture a big part of these differences, especially for age of first birth. And as a reflection, I think that this results suggest that genetic influences on fertility may be more important when social norms and economic conditions allow a broad range of life course alternatives. So we have the technology for being married, having sex, but not necessarily having children. Women might choose to have to postpone their fertility to school, to participate to the labor force. What can be relevant in fertility research is that with a change in social norms in terms of one is the best age to have a child, this leads to delay in fertility, but also leads to higher variability in the probability of conceiving. We know that many diseases or genetic that have a genetic basis, like polycystic ovary syndrome and so on, they are more relevant for women that try to have children later in life. So there might be women that can have children until their 40s, and women that have a more problem early on. So it would be important to study the genetics of the kids. Yes, but about the policy implication, this might show that some people react differently to change in the social environment. So the power of the pill may be stronger among people with a predisposition or preferences or attitudes, let's call it, or unobserved genetic that now can be observed factors for postponement, lower fertility and childlessness. So we might think that the introduction of contraception might have had a different effect across different. Yeah, thank you.
So, these fertility traits, the males and females may have different effects. Have you ever tested for the G by sex or G by pills by sex infection?
Yes, we decided to keep only women because especially I think the most interesting trait is age of first birth, and we don't have it for men. But we see that we did a bit of analysis with men, and we see an effect of the pill availability also for men, for age of first sex and childlessness in particular. But it's much lower than women. But this gender difference, I think it's quite interesting.
So besides the age at first birth, total number of children we have versus sex, do you think the length of the reproductive period is measure of the age at menopause minors?
Yeah, so this is not from this paper, but another one from the GWAS on fertility, and we look at genetic correlation with other traits. There's an interesting pattern that is so people who have genetic predisposition for later age menarche also have a genetic predisposition for later age for birth, for sex, and education, later education, menopause. They all go in the same direction. We're quite, and actually, some people even did with UK Biobank. They look at mortality. They look at age where. So now UK Biobank has some information on mortality of the respondents, but also has information on parental mortality. And so they did a GWAS on the age when parents die, and there seems quite strong correlation with all these traits. So there seems that there is a common genetic pattern of later in life, and so for example, the menarche and menopause is positively associated. It seems that there's a shift of both the traits. And also this, it's purely genetics or is other things, because we know that all these traits are associated with basically higher socioeconomic status. So it's interesting.
Super interesting. But to the first bullet point, so going back to what I was thinking about earlier, to be kind of convinced of that, I really want to be convinced that this isn't being driven by education genetics. Have you in that model where you did the robustness checks, were you interacting the education PGI also with the pill diffusion and the all in the same model, you know, because it could be that this is all being driven by education genetics, which is highly correlated with the fertility.
Okay, but that doesn't really matter what kind of PGI is.
Well, for that first statement, it does. Is it really genetic influences on fertility?
Because the thing is that the EA PGI and the age of first birth PGI are highly correlated. It's one of the biggest genetic correlation, these two traits, and we know that they both have their own specificities on the electric one. It's estrogen receptor genes and things related to reproduction and for the brain and so on. But how do you work when these variables are so
how highly correlated are they? I don't know.
Correlated.
Yeah, how highly correlated are they? I don't know.
Well, I don't know that. I know the genetic correlation is about 0.7.
Oh wow. I mean, I guess it would be interesting to see. I don't know if you put them both in the same model, then maybe at least they're unique portions, seeing what's driving it. Or I mean, I think it's also just as interesting to see if you see similar patterns with education, you know, some other PGIs.
Yeah, no, we had
Yeah, interesting. Also, I don't know how to place this or how to think about this. And it's just kind of is a lot of this pill diffusion and the uptake of it seems like it would be very much driven by religion too, religiosity and how much that varied by these different regions. And I'm not sure how to operationalize that or how to think about that in the model, but like, how would you, especially because I know the Catholic Church was so against, still is, against the use of contraception, but especially when it first came out. Is there any way that you can look at the religion of participants or?
I think from other data sets, probably we can see. I don't think that the local authority level, though.
Yeah.
That's but over time, that's we need to find things that are very geographically, but also over time.
Yeah.
So
Yeah.
every year. Participation to mass, maybe not.
And also another thing is like distance to university, like how deep was it for these women to go to college and to, you know, based on what they were, and that could be an interesting.
Yeah, we have another
ID.
project on that. We calculated for each one and distance university.
Oh, neat.
Yeah, data are a bit massive. So the different type of colleges and
Yeah.
another big change in this cohort is that the UK has this they call it a new university. So in the 60s they created a bunch of new universities and so people that have access to tertiary education. Before.
Thank you.
Yeah.
So, coming back to Lauren's point, I like, do you have a sense of, say, the average age at pill uptake by levels of the polygenic index? Because I'm thinking that those who are highly predisposed might be like more and you might just see the mean age to be lower, just or higher, I don't know.
Okay.
That
okay, so you would like to have a bit more into the mechanism on yeah. Also, this though changes over time, right?
Yeah.
So you should have some sort of analysis. But that's interesting. Yeah. Yes.
So I think you made two related points on the previous slide, on the limitation slide. One is that the polygenic scores are noisy, are proxies of the true underlying genetic risk. And then the G/E interaction coefficient is small. I wonder to what extent the second statement is due to the fact that the polygenic scores are not perfect.
Yeah.
And do you have a sense of, if you actually have the perfect polygenic score, how big the interaction will look like?
Ideally, I think it would be nice to have two polygenic scores calculated on two different independent samples. I use one as an instance for the other. That could be solved this measurement error problem.
You're using 1/3 of the biobank. It seems like you take 1/3 to use one PGI, 1/3 of each one PGI.
But then they are correlated. So if I use the other PGI, is still correlated.
We're not up on I'm not up on this Turley Becker point because I don't think we yet are convinced about this, or maybe
Okay.
Q is.
Right, it's related to sample size. If you have a low sample size, it is a big problem. But with the sample size of UK Barban, we see minimal impact on standard error estimate.
No, that was making my life easier. So if I could use the entire sample, yeah.
But I thought you said before that you could use it if you meta-analyze it. Am I not?
No, they claim that not even for meta-analysis.
Oh, okay. Understood.
Because I did all the regression for all the 3 samples, and I thought, okay, maybe I meta-analyze them. But yeah, you're not convinced about that.
I don't think we've read the paper you're describing carefully enough because I think Q's lab has done their own analysis in parallel with that paper to suggest that in big enough samples it's not a problem.
Right.
But I don't think we fully have integrated whatever the claim is in this paper. Is that fair to say?
Yeah, and it's a submitted paper, so we can send you a manuscript. It does have some empirical result, and we'll read the Becker paper again to see what exactly they did.
Yeah, I will be in touch.
Have you already done thinking about you're using typical PGIs that are predicting the level of the outcome, and there's maybe a new class of PGIs that would be targeted to explain variance in the outcome.
Yes. I haven't thought about that. Would you use them?
Well, I guess I'm asking the conceptual question. Like, if you had them sitting in your data file, would you prioritize one versus the other? I think a separate point is that Q's lab would probably be able to generate them and do all this analysis with the VPGS and VPGI type analysis. But the question is more conceptual about whether you think EAVGI or age of first birth or whatever, PGIs that are targeted at predicting the mean is what you want. Here, when you say, because the second bullet point is about social norms being the environment, and it's not clear whether social norms being the environment would be interacting well with the PGI as they're constructed.
And you actually did show the cohort effect on the variant phenotype, right?
Yeah.
So is it fair to also ask whether contraceptive pills or genetics will have an impact on the phenotypic variance?
Yeah, I mean, the cohort analysis came out after we got the first rejection of the paper. Then we thought of it. It's extremely difficult to convince the reviewer that exactly our measure is completely exogenous as a treatment, and we are very conscious of this. We know that it captures a lot of the things, but so we, in my demographic thinking, we went back to the cohort demographic cohort analysis, and we see that okay, this a change in cohort. If we divide by this new measure that is PGI, so we divide it by PGI, we see that there's a divergence. So this is a clear G by E interaction, and there is obviously something exogenous. It's a different. There's no PGI, but if PGI is a fairly constant across cohort. So we try to rethink the paper in how much this cohort differences can be explained by different measure of environment. And this is why not only the pill, but returns to education, so this is a bit of direction we try to explore with the paper.
So I actually wanted to follow up on exactly what Jason was just talking about and earlier what he was talking about, which is if you had the perfect PGI for fertility, what would that phenotype be? Because I guess there could be many definitions of fertility. My first thought was not the predisposition for postponement. I would have thought we were thinking about things like the likelihood of a person who wanted to have children to have children, meaning that they want to, so that's established. They're trying. Some women can do it, some women can't, and there's perhaps biological reasons why, or genetic reasons why they may or may not be able to conceive. But I guess my question to you, as you sort of alluded to in terms of your limitations on, you're really only capturing this proxy of fertility. What exactly would be the phenotype that you would rather be looking at?
Yeah, this is a good point. So we had a lot of discussion when the GWAS of fertility of measure of fecundity of individuals, or is it because it's so there were on one hand we think okay, it's impossible to measure exactly the probability of conceiving, and we actually was on this because then there would not be data, so we have this as a proxy. On the other hand, I think we are also interested on the behavioral part. So this is more like a life choices. So when people want to have children, how many, and so from more sociological aspect of fertility, and I think the pill has an effect on this side of on the decision. So I can wait until I wait for having a child, and especially among these cohorts, it would be interesting to look at more recent cohorts when many people start thinking of having children when 30, 35, and so on, and there probably the probability of conceiving is much more relevant. But yeah, I think this is more on the behavioral side, really, and so that's why I'm not sure that it matters a lot what kind of PGI I use. So even if I had I don't know, a combination of several, we were even thinking about, I don't know if it's a case idea to do like a factor analysis of different PGI and have a PGI that captures something.
You say it doesn't matter, is it because, so the correlation among the 4 or 5 PPI you might consider is, you said one EA versus one fertility ones, but childlessness versus age of first birth, what's
It's
0.5 orders of
higher the genetic correlation.
So partly what I take from James is that even with what you have at your disposal, you might have different hypotheses on the childless PI versus the number of children ever born, PI, in terms of custody, maybe. And therefore, the reactions to these norms may be different. Right? And you've already said, even based on the age of the person at the time, also making these decisions. Seems like what's stopping you from doing those things? Is it the sample size? Is it you've noted? You said that the PGIs are correlated and highly. Is that the
yeah? I mean, this makes the interpretation simple, and then we have one measure G, one measure E, and we look at interaction, and we try to explain the cohort variation that we see before. But yes, no, it's definitely something that we will definitely do with PGI.
Or I wonder too if you could regress out the EA PGI. If I could understand when they're so highly correlated, things in the same model might not. But if you could regress out the variation in the EA PGI from the fertility PGI, and so you're just left with what's not explained by education genetics and see what happens.
No, this like there is all these papers on the what did it call it, GSM, not the
genomic stuff?
Yeah. And that could be interesting to the
Yeah, it's almost like
Any remaining questions? Thanks so much for your time.
no, thank you. Thank you.
Oh yes. Yeah.
Important. Well, start with this. So let's see the time. So two initiatives important in Europe. This is the European Doctor Network on Social Science and Genetics. So we got a large European grant with all these universities in the Netherlands, in Germany, Italy, Norway, Sweden, and the UK. We will have 13 PhD studentship in these 8 universities. The program will start in October 23, and we will start to recruit this winter. And basically it works like this. So 13 scholarship in this universities, but it's a European cohort of students in which they have a common training together, and they will have individual training on the projects, and there will be exchange between universities. So if you are a master student, or if you know master students interested in doing this work, it's just gonna be exciting. So
The actual PhD would be in a variety of disciplines, or
yes, so the hours are both in the Department of Statistics, and we'll do something more on econometry. We have 2 projects, one is on genetics, the other on the econometry. So, yeah, related to genetics and social sciences, and then the specific projects, but we will advertise how to do it, but could be a nice experience for PhD.
So is this a specific thing students will have to search for, or do you just apply to separate institutions?
The way it works is that we have 13 projects that are quite wide can be changed. And there are some connection also with the institutions that are not universities. So it's all a requirement of the grant. So the basically one can choose the scholar or give a preference for a particular project or a particular university, and yeah, and I think it's going to be fun. And the other thing is that okay, yes, sorry to advertise again. So this is last year we had the first European Social Science Genomic Network conference. We took the same name of the network. We want to the two things go together. This is Bologna. This is a faculty of law. Actually, we had a conference there. Should I put other pictures of this room with the fresco or something? Well, I think so. Pietro and I, Pietro Birolli and I, that in Bologna, we got this funding, and we thought that it was about time to have a similar meeting of the IGSS also in Europe. Why Europe? Because there are many people working on genetics and social sciences, both in sociology, economics, psychology, and so on. And there are many data come from Europe, and so there will also an opportunity to talk to the data provider and so on. We did first in Bologna. We do it. They asked us to do it again in Bologna, and we said, okay, it was a lot of work last year. Probably this year will be easier for us. So we'll do again in Bologna. The tentative date is 18-19 of May. We have to secure them. You have to see the availability speakers and so on. And there will be a call for paper, possibly by the end of the year. Yeah. Thank you.